The attempt that we are trying to do here is a cell review, and this is a pretty basic review. I always have my students highlight stuff in the book. That way we can always agree on what is being tested. I'm glad you're here though. However, I am so glad that you're here, and so we're going to do a quickie review on the cell. Remember, what do we call the basic unit, fundamental unit of life? It is the cell. And so just for a few minutes, we're going to talk about some different kinds of cells, prokaryotes versus eukaryotes. Typically, we know that prokaryotes are a lot smaller. They don't have a true nucleus. They have a nucleoid region. They're not bound by a nuclear membrane as such. A lot of times they'll have something called a plasmid inside. There are a couple of different domains that I would like to talk about when I think about prokaryotes. I want to think about the bacteria and the archaeans, those. When I think about archaeans, these are bacteria-like microscopic thingies that can live in very extreme environments. We're talking about halophiles that can live in very salty environments. Methanogens live in methane environments. We could have thermophiles, ones that live in very hot environments. Plants, animals, fungi, protists, these are eukaryotes. Eukaryotes are typically more complex than the prokaryotes. A lot of times they will have membrane-bound organelles, and the prokaryotes do not. When I think of a bacterium, a lot of times there's a capsule on the outside. It makes a very sticky outer coat. I think that's how bacteria stick to your teeth. And then think about this. As these bacteria metabolize, they produce acids which dig craters in your teeth. What would you call that? It's a cavity. That's exactly right. Sometimes these plasmids that I was telling you about earlier that are found in a bacteria, they can be exchanged between cells. And right now I am actually studying in one of my graduate classes about the process of transformation, where parts of DNA can be transferred between cells, and plasmids are oftentimes important in that process. Remember, the nucleoid in a bacterium is just an area of DNA coiling. There is not a distinct nuclear membrane around the nucleus in a prokaryote. When we think about animal cells, one thing that is very unique to animal cells is a lysosome, and this literally means breaking body. Lys, break, soma, body. And so lysosomes break down worn out cell parts, and they also help to digest things that white blood cells bring into the cell and capture. That's kind of important. Remember that ribosomes are the site of protein production, and after protein production occurs on the ribosome in a process called translation, then a lot of times these proteins are modified and packaged in the Golgi apparatus, but before they are finally placed in vesicles. Remember, these vesicles can be used either inside of the cell or outside of the cell. If they are transported outside, that could be a process called exocytosis. If a cell wants to bring in, it is called endocytosis. Endo inside, exo outside. When we think about plant cells, remember, we think about the place where photosynthesis takes place. What do we call this? Everybody said the chloroplast? Chloroplast. We got it, Barnes? Yes, we're on it. A lot of times plant cells have a cell wall, and this provides rigidity in addition to the plasma membrane. And it's also what survives in a piece of wood or a plant after everything else dies. It makes hollow spaces, and in effect it makes the wood less dense than water. It makes it float on the top of the water. These cell walls are what create the little cells, cellae, as they were originally defined by a monk. Plants a lot of times will have vacuoles inside, and we do know that vacuoles can store nutrients, pigments, toxins, and things like that, water. One of the things that I really think is interesting is that poison ivy, the poison part, is inside of this vacuole. A lot of times you have to disturb it to release it. So kind of like the worst thing you can possibly do is decide that the large stand of poison ivy needs to go, and whip out the weed eater and start weed eating that. The only thing worse than that would be to decide that you need to burn it and put it in a fire, and all of a sudden now you have induced this toxin into a breathable form. So these are both really a bad idea. So if you're allergic to poison ivy, leaves of 3, let them be. Berries, white, poisonous sight, hairy vine, no friend of mine. Those are all things that you need to know about poison ivy, and it's contained inside of these vacuoles. Don't disturb this stuff. And definitely don't get the weed eater out after it. And then what's the last thing again? Don't burn it in a fire. You can get in some deep trouble with that. When I say deep trouble, I just mean some respiratory problems. That's not cool. Let's talk about the fluid mosaic model of the membrane just for a minute. There are 2 layers of fats. There is a hydrophilic portion that projects outward, and these tails are hydrophobic. They project inward. Lots of times there are membrane proteins that allow for molecules to move in and out of the cell. Listen to me. If this protein just simply allows stuff to move through it, it's called facilitated diffusion or facilitated transport. What if we have to have ATP to unlock it and change the shape to move something? What would we call that? We call that active transport. Everybody say active. ATP. That's how I link these together. I know if it's active transport, you got to have ATP to open it. And why is active transport important? Because a lot of times the body wants to move stuff against a gradient. That means move it uphill in a direction that's not normal. Now, let's talk about that for a minute. What is a normal direction? A normal direction is from high concentration to low concentration. That process is called simple what? Simple diffusion. Very good. There is another word that talks about water movement, and it is osmosis, usually movement of water from areas of high to low water concentration through a selectively permeable membrane. Is what I've memorized all of these years. Remember too, another thing that influences water movement in your body is sodium. If there's high sodium somewhere, water is going to move there. And so I always say, where Mary goes, the lamb is sure to follow. So Mary is the sodium, and water will always passively follow right behind it to dilute the sodium. Inside of the nucleus, there is the DNA. And the DNA is contained on chromosomes. Basically, this is chromatin wrapped around these histone proteins. This is another wacky doodle stuff. I need you to check it out. Apparently, it's not just your DNA that determines your health. It is how these histone proteins are locked or unlocked by methylation or acetylation. And a lot of times, apparently, the expressivity of our code is not always just the code itself. It's epigenetic effect. This is kind of a new thing to look that up sometime. Check that out. Remember that protein production begins in the nucleus. That's where the DNA code is copied in a process called transcription. Everybody say transcription. And then the code is carried out on a messenger RNA onto a ribosome. It might be associated with a endoplasmic reticulum, in which case the endoplasmic reticulum would be a what? An RER, a rough endoplasmic reticulum. And then translation occurs on the ribosome. And sometimes the protein will fall off directly into the RER, and it'll bud off a little package and then send that over to the Golgi for modification, and then a final vesicle will be formed from there. Remember that when we talk about chloroplasts, that chloroplasts use CO2 and water and the energy from sunlight to make oxygen and glucose for us. And then for us, we have mitochondria that break down the glucose or the fuel in the presence of oxygen to make water and CO2. So the one thing I want you to dig is that the output of one of these processes is actually the input of the other one. It's called the circle of life, baby. We got it. We're on it, Barnes. All right. Let's talk about cilia and flagella for just a moment. In humans, you might have cilia in your respiratory tract. You might have, that would be male or female. And apparently the ladies have cilia in the fallopian tubes. It helps create a downward current for the egg to move from the uterus down, from the ovary through the fallopian tube to the uterus. What's the only location of the flagellum in the human? What is it? Male sperm. That is the correct answer. Remember, we have male and female sperm. I want to talk just a little bit about something called extracellular matrix, and I need to go pick up a little bit about the chloroplast, and we're about done. Apparently, even around your cells, there is a collagenous extracellular matrix. Why is this important? It adds integrity, strength. We even have collagen in our blood vessels, and vitamin C is important for that. So vitamin C is important to help add structural integrity and skin, I would probably say strength, and maybe possibly slightly pliability, maybe not. Let's go back and talk about chloroplasts just for a sec. I want to remind you that there are discs inside of the chloroplast where this photosynthesis occurs. What are these called? Thylakoid discs, and they're in stacks of green poker chip looking things called grana. And then what is the liquid inside of the chloroplast called? This is called the stroma. In the mitochondria, there are little projections in there called cristae, and I haven't really talked about that so much. That was just something I seemed to leave out earlier. Good studying, A+100. Keep up the good work.